Well, a chromosome carries all the genetic information. So in every cell in our body, we have 46 of them, and they have very long DNA molecules. In fact, each chromosome has a very, very long DNA molecule in it. But these molecules are linear, and that means, of course, they have two ends, just like a shoelace. They have two ends. And a little bit like a shoelace that doesn't have the end of it protected, you know, the end of a shoelace can fray away, and the shoelace won't work too well if it frays away. And so similarly, the end of a chromosome can also fray away, and in fact it does. And what it has to have is, as we now know, a cap which protects the end. And when I started work on the tiny chromosomes that I was working with in Joe Gall's lab, nobody knew about what the molecular nature of the cap of a chromosome end would look like. So just being able to know what the DNA sequence was like, and it consisted of repeats of the building blocks that make up DNA, and in a very sort of repetitious, monotonous seeming way, you would have two of the building blocks, TT, and then another 4, GGG, repeated many, many times, tens of times at every chromosome end. And this was just totally, you know, bizarre. Nobody had ever seen something like this at the end of a DNA of any kind at all. And it was even stranger because it clearly wasn't carrying genetic information. This strange sequence couldn't make any sensible genetic information. And it was strange because from molecule to molecule, it became quite clear very early on when I started working with it that each molecule was not quite like the other. Some had more of these repeated little motifs of sequence at the end, and some had less. So there was a whole spread of different sizes of telomeric repeat tracts, as we started to call them, once I'd found out what the molecular nature was.
Well, discovering that the end of the chromosome consisted of 6 nucleotides repeated over and over again, certainly in one sense seemed to be a boring finding, but of course you publish your boring findings as well as your non-boring findings. Quite frankly, at that time, we thought that this sequence must have something to do with how these chromosomes were able to replicate their ends, how they solved this end replication problem. But we didn't hypothesize how it worked, and to be quite honest, we didn't even realize at that time that the sequence that Liz had discovered at the ends of these molecules was the same, or essentially the same, as the sequences at the ends of all DNA molecules of all organisms, including humans.
It did emerge that, in fact, this kind of DNA sequence was not just you know, a once-off that was invented by this pond scum organism, which would be very rare in biology anyway. But in fact, all organisms except bacteria have the same kind of DNA at the ends of the chromosomes.